Hello children, today we are going to study a very important technique in biotechnology, that is mass spectrometry. Many techniques we have studied till like protein fingerprinting, two-dimensional gel electrophoresis, protein sequencing, electrophoresis, etc. But this mass spectrometry is very special and unique. It is very useful because with the help of this technique we can obtain protein structural information and where the amino acids they are located and what are the types of amino acid. And the very important aspect which mass spectrometry is having, that we are needing a very little sample which is as little as picomoles, which is equals to 10 to the power minus 12. So you can understand other techniques they need where the sample in large amount. So somewhere if that we are having less sample, so with the help of mass spectrometry we can analyze the sample, but with other techniques this is not possible. In mass spectrometry we use the apparatus, and the name of the apparatus is mass spectrometer. Mass spectrometer is basically it is a analytical device that determines the molecular weight of chemical compounds by separating molecular ion according to their mass charge ratio. Most of the techniques basically that they separate proteins on the basis of mass, but here this instrument is going to take mass charge ratio. And why this is the basic principle behind the mass spectrometry, and this only this that property makes it so unique and so compatible to just separate different proteins. Later I will explain that how. Okay, first we will see that this is a layout of outline of mass spectrometer. Okay, so in mass spectrometer it is a very simple device. In this we are going to use the protein samples which are basically ionized and vaporized. Okay, and these ionized and vaporized samples, how we will prepare, that also I will tell later. Okay, so that vaporized and ionized samples basically that we direct inside the mass spectrometer. Okay, afterwards that with the help of electromagnet we are going to just direct it. So the red line is showing if that electromagnets are not there, so maybe the way of this these proteins it may be like red one, but with the help of electromagnet what we can do, we can just direct it to the amplifier. Okay, and here a vacuum pump is there to create vacuum inside it, and with amplifier a chart recorder is associated. Okay, and this chart recorder is going to just give us the results. Okay, so this was basic outline of the mass spectrometer. Okay, and as I was telling you all that we are needing ionized and vaporized samples. So how we can prepare the ionized and vaporized sample, we will see. A technique, the name of the technique is MALDI. The full form of it is the matrix assisted laser desorption ionization. Again I'm repeating it. MALDI, the full form is the matrix assisted laser desorption ionization. Okay, and we use to vaporize and ionize the protein sample, because the most of the protein samples they are generally non-volatile. So with the help of this technique we are going to make them vaporized and ionized. How? Like that we take sample, okay, then we convert it into condensed for that condensed phase, then with the help of solid matrix we convert it into gaseous phase. Okay, now that what we will do for ion formation, a pulsed laser beam we are going to direct on the sample which is suspended in, as earlier I told, a solid matrix, and the charged molecules of the samples will be created. Okay, so with here the matrix plays a key role, okay, in this technique by absorbing the laser light energy and causing the matrix material to evaporate. In the gaseous phase, the matrix plays a role in sample ionization. Okay, then these charged molecules are directed by electrostatic lenses from the ionization source to the mass analyzer. Means in the mass spectrometer we are going to direct these charged samples, where that with the help of amplifier the result will be recorded. Now I'm going to tell you an example which is going to show you importance of mass spectrometer. Why this mass spectrometer is unique, and it is very compatible for the that protein structural information. Suppose that we are taking a protein whose molecular weight is 20,000, okay, and it is having that 5 charge forms, 5, 4, 3, 2 and 1. Okay, just see here, suppose that if that the charge form 1 is there, so in normal case the molecular weight will be 20,001. Okay, but if we will take mass charge ratio, so it will be is equals to 20,001 upon 1, which will be equals to 20,001. And if we will see the charge state, means where the two charges are there, so in that case the molecular weight will be 20,002. And when we will take mass charge ratio, so it will be 10,001. Same in case of 3 charges, it will be molecular weight will be 20,003. And if we will take mass charge ratio, which will be is equals to like 20,003 upon 3, which is equals to that 6,668. Okay, now 4 charges, the molecular weight is 20,004 and mass charge ratio will be 20,004 upon 4, 5,001. Then 5 charges, so the molecular weight will be 20,005 and mass charge ratio will be 4,001. So you can see that if any of the technique which is going to separate these forms of proteins, okay, all 5 forms, so that technique will not be able to detect all of them as separate protein. Why? Because there is only the difference of the molecular weight is 1, 2, 3 and 4 and 5, only 5 digits. Okay, 20,001 and 20,002 or 20,003 or 20,004, 20,005. Any of the that technique is going to detect it as same as 20,000. Okay, because not much difference is there. But when that this mass spectrometry takes it as mass charge ratio, because the principle behind that is mass charge ratio. So what see the difference between that all forms of protein is very big, like 20,001, 10,001 here, 6,668, 5,001 and 4,001. So the mass charge ratio will detect all of the forms of protein as separate protein. Okay, so we can separate them on the basis of with the help of mass charge ratio, not with the help of the molecular weight. So for all these cases it is very important to use mass spectrometer, because other techniques it will not helpful for us for separating or characterizing these protein. With the help of mass spectrometry only we can characterize them. Thank you.